Microsoft. I am professor at the University of British Columbia, and I'll give you a presentation today about the deep docking software and approach we've developed in the last couple of years. And by default, by the name of it, you can realize it has to do with the docking, which is one of the most widely used virtual screening tools out there. I'm sure most of you, if not all of you, are using it. And we are not exception. We've been using docking for decades. And here's a typical example of a workflow of docking with the target as an antigen receptor here, which is a dimer and it sits on the DNA. It's a transcription factor. And so, as in many other conventional workflows for drug discovery, we would take that protein apart, we would look at the fold composition, we would build surfaces, look at the atomic level resolution, then we map all kind of properties on the surface and run docking. And there is no shortage of programs. To my count, there is probably close to 100 of them. So different people use different ones. And just like you heard from the previous presentation, there are several of dozens of most conventional approaches. So we use different docking tools and just at the end of the day to find a molecule which fits in the target site. I'm sure you also know that this success rate of this exercise is around 10%, 10 to 15%, but it's still much better than 0.01 or 0.03% when you do just random virtual wet lab screening. And that was the status quo for a very long time. So we've been doing this type of docking-based drug discovery for decades. So we would normally start with 5-10 million molecules, a virtual database such as Zinc database and many others. Then we would run some docking exercise to identify 5 to 10, 50 to 100 sometimes candidates. We would screen them, and then wet lab assay would confirm if there are actives. And again, as I said, typical hit rate would be 10-15%. And so that's how we start a real drug discovery campaign. Again, that's been going on for a very long time. It was a very successful approach and many people using it. Ourselves, we've been pretty successful in doing so. And 5 million molecule was enough for us to start various rather successful drug discovery exercises, and everything was working. But something has changed roughly after 2015. And that is the number of available chemical molecules. So the size of zinc itself grew from 10 million up to whatever it is, 1.4, 1.5 billion now in just under 5 years. So basically the amount of chemical information just exploded. There are various reasons for that, including the emergence of robotic synthesis, combinatorial libraries, and click chemistry. But bottom line is that now 10 million molecules to be docked is really a drop in the bucket. It's really, we just roughly, just basically scratching the surface if we only go for 10 million molecules. So available chemical space right now is sizably larger. And as I also know that there are already larger libraries, such as in the mean real space, going to 40 billion molecules. And in principle, the possible number of molecules, as we know, is anywhere around 10 to 60, 10 to 100. That's a high ball estimate, or 10 to 20 power of 23, which is more or less synthetically feasible compounds. So again, all of a sudden, if you would just go into even conventional zinc database, which is 1.5 billion molecule, and try to dock it as you used to, you're not going to go anywhere. So this is a problem. 2015 brought this significant challenge that currently available purchasable chemical compounds no longer can be docked using reasonable amount of computational resources. And so that's when, roughly in 2018, 2019, my lab went to a very old original idea, which I published in, was it 2006, which was called progressive docking. And back in 2006, I said, what if we stop generating docking scores? What if we start predicting docking scores? Because you can dock just a fraction of a database and train machine learning approach based on the real docking scores and start predicting. And this progressive docking was basically predicting compounds as we go through the database, as we dock through. It starts predicting which compounds which haven't been docked yet, but don't look very good. And so you start progressively removing them from the remaining docking days. And so linearly, if you compare, computational time required to recover a docked molecule. Obviously, it's linear. But if you start progressively removing bad molecules from the remaining undocked molecules, undocked docking base, you start speeding up. Again, it was 2006, but it wasn't really a really practical exercise at the time, because I wouldn't remember exactly, but I'd say the variable docking database sync was probably, or I don't even sure, don't remember, sync was around below 1 million molecules. Maybe our docking base was 500,000 molecules. So in other words, you can actually dock 500,000 molecules using reasonable computational cluster, which was available back in 2006. But now, removing unwanted molecules or augmenting enriching database for potentially good ones becomes a much more interesting problem, because now computational resources, again, don't match database size. So we revisited this 15 years old concept back at the time, and basically started predicting docking scores. So you would take your molecule and then you break it into some sort of chemical descriptors. It's pretty standard chemoinformatic exercise. Fingerprints lately become pretty useful. And then you would feed that in deep neural network, and then you use real docking scores as you go on the database as a label. And then you start approximating undocked molecules using QSAR, basically chemoinformatics approach with deep model. And then your workflow becomes all of a sudden 100 times faster. So by using this approach, we were able to augment the docking protocol, and we could dock 1.4, 1.5 billion molecules now in about, well, using our computational resources in 3 weeks, while it would take us 2 years or so if we just do it conventionally. So we have pretty good cluster, probably several thousand CPUs and GPUs, and using Glide, which is good but relatively slow, 1.4, just conventional sync again, would be 2 years. Instead, we run deep docking when, again, you putting this AI module on top of docking protocol, and things become 100 times faster at the cost of about 10% of actives, or we would call hits, being lost. But this is a good trade, 10% for 100-fold accuracy. And then we ran this approach on the family of most conventional drug targets from the literature. And you can see that the enrichment of a database for different targets was reaching up to 4,000 folds. So basically now the process becomes up to 4,000 times more efficient. And so you see that we run it iteratively. So you can do it in one shot. Obviously, you can just, dock million molecules from 1 billion and predict the rest based on the machine learning scoring function. It's no problem. But just to preserve a bit more actives, by actives I mean docked molecules in this case, we run it in iterative steps, and we figured out that about 10 to 11 docking iteration steps is sufficient. Of course, the first drop is at the first step very significant. So sync goes from 1.4 billion to roughly 300 million. And then iteratively, or as we would call previously, progressively, it gets reduced. So at the end of the day, you actually dock only 1% of molecule, and 99% just get predicted based on your deep learning model, which again becomes progressively more accurate. And here we augment the training base by predicted actives rather than removing predictive inactives as before. And so your model becomes more accurate, less greedy, and your database gets down to the remaining 20 million molecules in 10 iterations from 1.5 billion. And it was just on time when we finished this, when we had a paper ready, when COVID pandemics emerged. And so it was on time for us to look at the most conventional drug target now in COVID, which is main protease. In principle, there are 25 potentially druggable proteins in the viral genome there, potentially druggable 25 sites really. And so we selected main protease, which is also 3CL protease, as it's also called. And first time ever, we actually, as a report in literature, we were able to run the entire 1.4 billion molecule from zinc. So we ran our deep docking, which was implemented on GPU Autodock. We worked with NVIDIA in that project together. And so we ran that 1.4 billion zinc. And as you can see, this is the distribution of the average docking score. Here is just a random when one would use just Autodock Vina or auto-dock GPU or glide. That's kind of range of a docking score you get out of sync on random docking on 3CL Pro. And this is known protease inhibitors on a training set. At the time we used SARS-1, obviously we didn't have SARS-CoV-2 at the time. And that's where deep docking enriched for the docking scores. You can see this group of scores is significantly shifted toward negative side. So deep docking, in fact, enriched very, very significantly for best binders. So then we identified 1,000 potential molecules, and we published that paper as early as in March 2020, just at the time when pandemics was actually declared. In fact, preprint was published in February. And out of those, we ordered about 600 molecules and we screened them, and we found about 30 actives, so hit rate of 5%. More importantly, our work really set up the first ever reported billion molecule docking campaign, and a lot of other centers and groups followed immediately. So Oak Ridge Supercomputer Center, they threw in 45,000 cores, 45,000 GPUs, and docked physically 1.4 billion in one day. European Center with 27,000 GPUs did it in one day. More or less comparable what we've done when people used 8,000 cores, were able to dock 1.4 billion in 4 weeks. We used 5 weeks, but we only used 60 cores in that particular pilot study. So you can see the difference. And but more importantly also is that we cannot just use docking faster. Also, this speed enables us to use multiple docking programs. So at the next step, we decided to raise the bar and did the next challenge. So we ran the same pipeline, not for 1.4 billion, but for 40 billion molecules. And here you see the heat map, how 40 billion molecules have been docked with 5 different programs. So here you can separate actives. Again, by actives, I mean well-docked molecules in blue and red. So more or less after multiple filtering, you see that the picture of 5 programs probably produce generally half and half actives and inactive. But since we use 5 different programs and all kind of different tools, because again, deep dock enables you not just to dock more molecules, but to dock more molecules with many different ways. We started building different consensus protocols, and you see things get much, much bluer as we start congesting the database down to 40 hits. So we went basically from 40 billion here to 40 here, and obviously they're all blue. Anyway, it was a very interesting exercise. We tried 28 different strategies, different consensus strategies. And as you can see, here's the clustering of finally selected hits. So at the top, the agreement is reasonable, but in general, even you use the same 5 programs, you just, exercise them in different combinations. The consensus is relatively poor. So we had to explore it. And we ordered another 1,000 molecules and ran standard screening against main protease. And also we use live virus as well to screen them. And in this live virus, we would identify compounds which don't just inhibit the main protease enzyme from SARS-CoV-2, but also we would see how those compounds stop infection of cells and even lung organoids. So we had a pretty reasonable pipeline. And so out of, I don't remember exactly, 1,400 compounds we ordered from 28 different strategies, here they are. I see here, 2,500 we ordered, 1,400 we were actually able to buy, sourced from vendors. And of those, we found 162 active molecules, 162 main protease inhibitors. And this also enables us to assess how different consensus protocols make it possible to select molecules automatically. Because again, very important process here. If you get very good congestion for actives here, if you really squeeze your data very, very hard, then in principle, this process can go automatically. You don't need the human expert. You don't need to look at the compounds you predict because of such a rigorous processing of your data. And that was our approach. So we really wanted to establish automatic pipeline. We wanted docking to be human-free. And out of these strategies, only the two last one were expert-based strategies. And in general, again, the success rate here was more or less reasonable. As you can see, there are 28 strategies which all gave reasonable number of actives. Should mention that expert intervention still makes it better. So last two lines here, 12 and 19 hits, that's when a really experienced drug discovery person was looking at the results and handpicking compounds. But nonetheless, all other hits out of 162, 120 plus were generated fully automatic. So deep docking, as I will reiterate, not only enable us to dock 40 billion molecules, which hasn't been matched yet in literature, size-wise, but also enable us to develop very automated protocols, because when 5 different programs agree, your results probably going to be reasonable, probably going to be rather reliable. And you can see, again, the hit rate was very, very good. Again, close to 12%. This is very much the state of the art right now. And it's all automated. And it was also very, very handy for us, because our idea at the time when we found hundreds of actives against COVID, against SARS-CoV-2 virus, we wanted to build some sort of a bug defense system, what we call. We built a viral defense system. Because back in 2005, actually, we put together a grant 20 years ago, when we said that, in fact, the SARS-like infections can occur. And this is an actual snapshot from the grant we were awarded by Genome Canada, that we said that we need to build a knowledge base and computational facilities in case that new infections will emerge, so including previously unknown ones. And so we even mentioned HIV and SARS there, among the others, and West Nile and E. coli. And we said that we have to be ready for future outbreaks. And so with this deep docking platform, with pretty much automated platform, now we're trying to build such a virus defense system in case that more infections will emerge. And you start hearing now about, monkeypox and all of that. An idea is that this deep docking will be in the core of an integrated platform when you start with genomic information on the new virus and protein-protein interaction information, which is very important. So you generate all the proteins, you run them through functional assays, and you run proteomic analysis on interactions. And then you see the keep layer, and then you pass them into cryo-EM, NMR, and X-ray facilities to find the structure. And that's when then you run your deep docking, your other automated drug discovery platforms. And then, of course, you can advance potential hits into leads and the clinical candidates, do pharmacokinetics, pharmacodynamics, and on and on and on, in vivo and clinical trials. So this is a bigger dream we're moving toward to. So the deep docking will become hopefully the heart of this virus defense system pipeline. Again, this is a work in progress, well, pretty fresh stuff. And of course, we wouldn't be successful without a lot of support and collaborators. So I have to acknowledge a lot of people in my lab, people in medicinal chemistry lab and structural biology lab, a lot of virologists which have been testing our compounds in enzymatic assay, in live virus assay. And of course, the work was supported by a lot of funding bodies and agencies. So that will conclude my talk, and I'll be happy to take any questions.
Thank you very much for the interesting talk and perfect timing. We already have 2 questions. The first one is from Vladimir Sulimov. After you select about 600 compounds for experimental testing, 30 demonstrated activity against main protease. All these inhibitors were non-covalent? It's a question. So whether those were non-covalent?
That's right. So all this docking was non-covalent docking. All those inhibitors are non-covalent inhibitors. And that was our angle, because all of the other existing ones from Pfizer and Merck are covalent and pro. And covalent drugs, they have own pharmacokinetic challenges and toxicity challenges. And it's no surprise that even Pfizer compound is prescribed in combination with cytochrome inhibitors. And even with that, the pharmacokinetics is not great. Again, it's a good first line of defense, but in principle, we are looking in the longer term solutions, and we're looking for non-covalent ones. So that's our difference.
Thank you.
Okay, thank you. And the second question is from Dmitry Solodkin. Different docking protocols versus different X-ray structure. Should both approaches be applied? Did you perform exploration of X-ray structures?
Absolutely, that's a very good angle. Yes, so deep docking basically allows you to pass more molecules faster. And that could translate into more docking programs. And indeed, that could translate into more crystal structure, or better yet, I'd comment, to more molecular dynamic snapshots. So in principle, if you start bringing in more flexibility of active size captured through MD trajectories, then yes, your accuracy should in principle go higher. Or in fact, you can start using flexible docking. So at least at the final stage. So one of the filters you can start adding, you can start docking flexibly. Again, deep docking will enable to do it fast. But yes, so there is really no limit how you do it. Main thing is now deep docking is pretty much from the literature I see as people start doing things.
Okay, another question is about is from Igor Polikov. Did any of the predicted main protease inhibitors made it to an actual drug?
So we're working on that. So we have probably 3 good leads now going into further screening. So they're going through in vivo right now. So there is a decent PK/PD. Again, whether they'll make it into real drug, that's more regulatory question. As you know, the R&D, research and development phase of drug discovery is a year or 2. But the main thing is clinical trials and all of that. And even though for antivirals it will be fast-tracked, still it will take some time to run it through proper FDA or other health regulatory agency approval. It's not up to us. So as a scientist, we can provide something we think is reasonable, and then that's up to governments to make drug out of it. But so far, there are several molecules which are on good trajectory. I could also mention that even though these are non-covalent drugs and they go well into animals and they go well into human organoids and they kill virus well, we're also trying to enhance some of them with the covalent warheads, just like Pfizer and others, to push it even further, because if the dosage will be lower because of that, then pharmacokinetics could be better anyway. Again, these things are all ongoing, and if we can come up with something before the next wave of pandemics, if it comes, then that would probably make it in the clinic. I should also say another angle is that we're making this drug for free. So once we have a drug, we want to make it available to the world. And so in principle, if that will enable fast tracking in the data, sure, that will be drug.
Okay, thank you very much. Well, we don't have any questions in the chat. Okay, here is another question from Vladimir Paroykov. Since you have an experience in drug repurposing against SARS-CoV-2, have you estimated the perspectives here? I'm sorry, how you estimate? Yeah.
Yeah, so we've spent a lot of time looking into repurposing, as anyone else. So the first wave of drug discovery, obviously, was to find something which exists and approved and see if it kills the virus. And first few months, that's what everyone else and us were looking. And we actually published a couple of reviews on this. One is in JChem and others, Chemical Society reviews a year back, when we reviewed all those efforts. So in my account, nothing worked. So the only real example, more or less, was remdesivir, which was developed for MERS initially, or even for Hep C. And that was then administered intravenously through the clinics, and the course was like $20,000, and it worked somewhat. In my account, everything else, ivermectin, all the kinase inhibitors, you name it, all those compounds, I don't even remember, the gazillion of them all were suggested. None of them were actually approved, and none of them were actually used in the clinic. So repurposing for SARS-CoV-2 didn't work, unfortunately. There are reasons for that, but it didn't work in my account. So whatever works now, Merck and Pfizer, those are made particularly for this virus. Even though they use the old data, they use data for SARS-1 and MERS, but they did modify the compound. So it's not really repurposing. It's sort of tweaking, make it work, but these are new chemical entities.
Thank you. Another question. Can you solve the other task of screening many thousands targets against a few compounds?
Reverse docking, sure, it's no problem. But in my opinion, you don't even need deep docking for that, because you can even use conventional tools to do so. Different question is the utility of that, how useful it is to do reverse docking. In my account, in my experience, it doesn't work very well, because docking is not really a tool to find true actives, true positives. Unfortunately, I wish that it is, but it's not. Docking tool is to remove true negatives. It's when you get 1.4 billion, you have to remove 1.39 blah, 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 billion, which is not worth your attention. And then you focus on the rest. And even when you see even 5 programs agree in 28 different ways that this is a hit, our hit rate is still 12%. So it means that, in fact, if we reverse the logic, 88% of the time we're wrong. So we throw in, thousands of cores, computers, we throw in 5 programs, we throw in human expertise and 2 months of hard work of 20 PhD scientists, and 88% of the time we're wrong. So unfortunately, that's the state of the art, and that we can discuss what we want, but that's what it is. So using that inaccurate tool to map small molecule to biological effect through docking in thousands of proteins, it's not yet practical exercise. It will be one day, but not now.
Thank you.